Hey guys, today we're going to be talking about neurons. So these series of videos that I'm making are going to be completely everything you need to know about neurons, of how they function, the resting potentials, action potentials, grand potentials, how these signals or these electrical signals are carried through the axon from one neuron to another. We're going to talk about the specifics of each of these processes step by step. So this video is designed to be as an overview, as an introductory to what I'm planning on covering. So this series of videos is going to be good for students who are taking an animal physiology class or even a human physiology class. The processes are very similar. Or if you're taking any kind of higher level biology, this will apply to you as well. So starting out, this is a neuron, pretty obvious. So just a few structural things to consider. When we talk about the soma is like the head of the neuron. And within the soma, we have what's called the nucleus. That thing right there. And then we have these extensions that come out from the soma, and these are called dendrites. Now, what the dendrites are, they're actually going to be serving as the input into the neuron. We're going to talk about these things much more in depth later on, but these inputs are going to stimulate the neuron and therefore trigger an action potential. Just to briefly talk about what an action potential is literally just like it sounds. It's the potential for something to happen. It's the potential for an action. Now, not all of these stimulus or stimuli is going to trigger what's called an action potential. There's a specific mechanism involved in it that we'll talk much more in depth about later on, but I just want to put that out there. So, and this extension from the soma is called the axon. Now, within the axon, you can see that there are segments. These segments are covered what's called by a myelin or myelinated sheet. Now, what a myelinated sheet is going to do to the axon is going to help increase the efficiency and the speed of the signal being transmitted through the axon. Now, you also notice that there are little nodes that are not covered by the myelin sheath. Now, these nodes serve a very important function, and they're very critical to action potential propagation, which I will talk more about in depth. But these nodes are called the node of Ranvier, and these nodes are the actual sites of channels that open and close that trigger action potentials and propagate action potentials throughout the axon. And then over here we have the axon terminal, where vesicles are located that contain neurotransmitters, and then they are released into the synaptic cleft, which is the space in between one neuron and another neuron. This is how neuron to neurons interact, or neuron-neuron signal and transmission occurs. And we'll talk about that in depth as well. So this area right here is called the synapse. And one thing I want you all to know is that the neuron that is sending the signal, so in this case, this neuron right here, an action potential triggers, goes down to the axon. So this axon is sending out this signal to the next neuron. Therefore, this neuron is going to be called the presynaptic neuron. So whenever you hear me talking about presynaptic and postsynaptic, that's what we're referring to. So the sending neuron is the presynaptic neuron. And the one that is receiving that signal is called a postsynaptic neuron. These are terms I want you guys to familiarize yourself throughout this series of videos. Now, the way we're going to approach this, we are going to first start from the axon hillock, the site of where an action potential is first triggered. And then from there, we're going to talk about how the action potential is triggered. And then we're going to talk about the actual molecular mechanism that is involved in an action potential. What causes it? So an action potential is a depolarization effect. Once again, don't freak out. This will all be explained in the next series of videos that I'll be uploading soon. So we're going to talk about how sodium and potassium play a role in these action potentials, what they do and how they do it. That's something that's very important throughout this entire series of video, the interaction between potassium and sodium leaving the membrane of the neuron cell. We're going to talk about what it means to have a resting membrane potential, what depolarization means, hyperpolarization. But one thing I want you guys to start thinking about now is that there is a difference of charge between what's inside the cell and what's outside the cell. Therefore, it's called polarization. polar means it has two sides, or it has two directions, or it has two different charges. So you typically inside the cell will be a negative charge, and outside the cell will be positive charge. Much more in depth about that later, don't freak out. And then after that, we're going to talk about the mechanism of propagation of the signal. So once the action potential is triggered, how is it going to be propagated down the axon to the axon terminal? And we're going to talk about why it's unidirectional, meaning that the signal only goes in one direction. It goes from the axon hillock and it goes to the axon terminal. It does not go the other way. And we're going to talk about why that is the case and the mechanism involving that. From there, we're going to go to the axon terminal. And then we're going to talk about the mechanism that's involved that causes these vesicles containing neurotransmitters to be released and sent out to the synaptic cleft. And then from there, they can be picked up by the postsynaptic neuron, by receptors and other mechanisms that we can talk about, and how signal is transmitted between one neuron to another neuron. And then, once that is, we're going to come back to the site and we're going to talk about what factors or what inputs or stimuli are going to trigger an action potential. And we're going to talk about what it means to actually have a potential of action. I think that is all for now. Just be on the lookout for the next series of videos. Thank you.